All right, welcome everyone today. Glad to see everybody. Today we'll be working on our RNA Workshop 3, which is a continuation of the Workshop 2. First we started out with the RNA Workshop 1. It was in the bulk RNA seq, and now we have moved last time to the RNA Workshop 2, which we did a great survey, kind of moving through just your raw counts to be able to do your basic cell types. And we have our cell types clustered, annotated, out there, ready to go. Then our next step is to move down and do more of our downstream analysis. So we've kind of done a lot of the hard part, filtering the data, getting rid of the doublets, and getting our clustering resolutions correct, and our UMAPs and all that stuff, annotations. That's a lot of the battle. Getting those clusters and annotations right. If you don't get those right, nothing works after that. So that's a really important way to look at it. So it's very important. And then today, we're basically taking that data and we are moving to more of the biological interpretation and downstream analysis. Last time was more about identifying the cells. What are the cells? Now we know what the cells are. And today we ask the next question. What are the cells actually doing, right? And a lot of these things, the analysis pull back from really the bulk RNA-seq. We're just applying them to a higher resolution single cell, where if you look at like differential expression, we use things like DSeq2. Now we're going to use things like Surratt's find all markers. Pathway analysis, we're going to do FGCA the same, just instead of full ranked differential genes, will be cluster specific, right? Pathway scoring, instead of per sample, it'll be per cell. NMF, sample by gene, it's going to be cell by gene. Very similar, right? Cell communication, trajectory, other things obviously can only be resolved by using, the single cell methods. But it's really not a lot of the very same like theories and philosophies of how to do a lot of this we've already gone through. So today we're going to identify our cluster, marker genes with find all markers. And then we're going to run a suitable differential expression to understand how to do that the right way. We're going to go ahead and execute pathway analysis with FGCA and GSVA on single cell data, discover NMF programs. Then we're going to use add module score to score T cell functional states and also myeloid polarization. And then we are going to project hallmark pathways onto any cell set, all the different cell subsets. And then we're going to do two great downstream analysis that you often see, a trajectory analysis with slingshot and trade-seq for the differential expression of genes along that pseudotime. And then we'll also map cell-cell communication with cell chat and triangulate kind of the ligand receptor interactions, that are interplay between the expression and pathway evidence, right? And then finally, we'll show how to integrate multiple samples, and you can do more robust cell type composition across different patients. So just getting started, we'll install all of our packages here, pretty much the same stuff. And here's the thing for cell chat if you don't already have that. Slingshot, trade-seq, or bioconductor, and RCPL to speed up, and NMF is just a CRAN package. And you can kind of see here all the different packages listed. You can kind of look through here with their annotations and everything. And so the first thing we want to do is just reload that object from last time. If you go back, if you remember, we saved this object long TO9 session 1 annotated.rds. We're going to pull that back in. That's all I'm doing. It's just reading this file back in. And we'll see here that 3696 cells is just our object from the first field. And we'll go ahead and plot it just to kind of remind you of everything. Remember, we had the T cells here, NK cells, mast, B. We have our tumor epithelial down here, plasma cells, and endothelial cells there, as we see. And then our myeloid compartment here. Remember, we did everything. We had our macrophages, right? Monocytes, kind of this macrophage split here. And then we had our copy number, right? Where we were able to knock out we know this is aneuploid down here. And everything else is probably going to be mostly diploid. There's just a few weird things going on, artifacts. If you want to go back to the last lecture, you can learn more about those. And here's kind of just a general idea of our composition. Where if you remember, we had mostly T cells and some kind of macrophage monocytes that take over the population. So one thing we're going to go do before we move further If you remember, we annotated the cell types in 3 different ways. We used a marker panel. We, basically our own annotated genes. We used a single R with a reference data set, and we used the real published labels. So they put a lot of work into those labels, so we feel pretty good about those. And our annotations were almost pretty solid. I mean, we could have gone a little more in depth on some of them, but overall they looked pretty solid, except one place that we didn't like, didn't look good. If you remember the dendritic cells, the macrophage cells. So let's go back and look at that again and see what we see in the UMAP. If you remember over here in the corner, we have this weird macrophage population over here. It doesn't really match what's going on here, and we always kind of worried about that. And we looked at the manual annotation, the published annotations. These were actually all dendritic cells. And we went back to our annotations that we used, the cell signature we used to actually find these cell types. We noticed we forgot, or we left out dendritic cells. We weren't as comprehensive as we should be, or otherwise we probably would have caught those. And since these cells share overlapping markers with the monocytes macrophages, that's why they were labeled as macrophages. But that doesn't mean they are. It just means this is how complicated it is to do this. And then we found looking further was accurate dendritic cells. So what we're going to do is this is totally normal and the way things are done. This is not cheating. We are going to relabel this population, this cluster, to dendritic cells. And this is how it works. You do your first iteration, you learn more. Second iteration, you learn more. You read more papers, you find out, and that's how you figure. Sometimes you have to go to find out to be able to annotate some of these things. It's not obvious. This is hard work. This is at some level an art form and another level of verification. So there's nothing wrong with doing that. So there's 2 things we're going to do. Very simple. The first is we're going to change, I just mentioned, that macrophages subtype to dendritic cells, and we feel pretty justified in doing that. And the second, we have this macrophages monocyte derived label. It's just too verbose, and some of the problems later, like some of the programs later, like cell chat, won't like that. So we're just going to take that out and label it just macrophages. So if you look at the new one, very simple, not much different. We just labeled this macrophages and dendritic cells. You can already see it looks much more beautiful. Maybe it's the colors, but it looks much more better. That's really great English. But it looks great, I think. I feel really good about this. We could have split the T cells a little bit, but we already did that. We could have projected those T cells back onto here, but I want to keep it macro here for now. And I'm actually really proud of how well that worked out. And this just kind of changed up the distributions just a little bit, no big deal. And just to kind of remind you how to subset off different clusters, things like we did with the T cell compartment last time, because we'll do this a few times later. I just want to make sure to remind you is basically if you want to choose a cell type, you basically just set the identity up to a cell type here, and then you just do subset your object identities equals the cluster name. So T cells, tumor epithelium, we want both these clusters. We want just the aneuploids. Really you can just choose any column and what it is and pull it out, right? Choose the metadata and the column in the metadata, and you will pull out all the cells that are associated with that metadata, right? For example, T cells. And that's how we're able to subset out those all. All right. So now that we have our guy, our object back reloaded and everything's set up, our annotation is set up to go, we're going to move forward to our differential gene expression and find genes that define each cell type. And this is really the foundation of every downstream analysis, right? And we'll use 3 complementary approaches to do this. First, we're going to find the cluster markers with find all markers. Now, there's a lot of ways you could do this. I mean, you could probably take each cluster and do DSeq, things like that. But when you have a lot of clusters at once, find all markers is a really good function within CSAT to be able to find your cluster-specific differential genes. And it works really well. And to do that, you set your default object to RNA, not the SCT, because you want to do that basic normalization again. So basic RNA, we do our normalization, and we do our find all markers here. And if we don't put any clusters in, it compares every cluster to every other cluster. And we want only upregulated genes here. You can obviously put false and have both. We want genes to be detected in a greater than 25% of cells and a minimum log 2-fold change here of 0.5. And we're going to use Wilcox for the test. Basically what it's doing is just doing a Wilcox between all the clusters and finding out differential genes in each one. You can also use t-test here, much faster, but not as reliable. If you wanted a quick look, but Wilcox is quick enough. If you wanted to be to be more sophisticated and more in depth, you could use MAST here, and it basically models dropout, things like that. It does a better job of doing this in with single cell data, but it takes a little bit longer. It's a little bit more bigger, so we wanted to use Wilcox here. We'll use it later for our cluster comparisons, just to show you the difference. But here we go. We run that, find all markers, and it found 6,500 significant markers among those 10 cell types. And we can look here, we can post just the top 5 by log fold to change of each of those cells. So basically what we do is we grab markers per all of our cells, and it gives us an idea of what is located in each cell. One thing that's really important to see is the percentage in each cluster, because some bleed over. Like in macrophage, like CCL18 is found pretty much in both of these. It won't be that useful a marker, and some are specifically found in one or the other, like 84 here to 0.01. That's going to be a pretty solid B cell marker. So you can kind of see that, and we can go ahead and plot those out on our chart, and you can see here all we've done is just plot our genes with the expression. And we can see that these blocks are we want to see these very defined blocks for each one of them. These look pretty good. They're not the most beautiful ones I've ever seen, but they're pretty solid. You feel pretty good about them. And you can see some bleed over with each one, and that's okay. It's just a measure of what that gene does. Some of those genes are involved in some of those patterns, and it's not always easy to find genes that are only involved with certain ones. So anyway, this is how you can kind of get a good visualization of how well those genes how those differential genes split out into each of your cluster. And if you kind of look more into it, you can kind of look at some of the ones that did a better job and other ones though. But it all looks pretty good. And here's just another explanation right here of the different methods we could have used to run that. All right, so now that we've kind of found our cluster markers, what if we want to look at just 2 specific groups? Like let's say the tumor cells and the T cells. We just want to see the difference between those 2 only, right? Really get that difference. You could also do that as well too. And for this comparison, we're going to switch to MAST, just because it's a little bit more conservative and appropriate when comparing 2 specific groups. And it's really the publication quality one that you want to be doing anyway, to be honest. And so once again, you just changed your assay, right, to find all markers. But instead of having blank, now I put 2 different populations in, the 2 I want to compare. And I added MAST instead of Wilcox for here, and I was able to get my results here, and I'm just going to go ahead and plot it up here, and you can see it worked pretty good. This is tumor, this is immune. If you kind of look up here, you'll see a lot of these surrogate proteins. This is AT2-luad. You can see things like NAPSA, FLOR, MUC. These are all supporting, this luad cancer, this lung adenocarcinoma origin, and the left side is just all T cells. So this actually worked pretty well. And you can kind of look into it and feel pretty good about it. So that's how you kind of find your differential genes in different clusters, and that's how you compare to specific clusters within your sample. Now, if you're working within your sample, find all markers is a good method, and it's totally okay to use. However, you cannot use 5 all markers and this similar kind of comparison across samples. That is a big no-no. And probably the most important thing you can learn in this workshop, and things that happen all the time. People are still publishing papers like this. It's crazy. You can't do that. There's pseudo-replication. By doing that, here's the thing. If you're comparing the same sample, all the cells there's no batch effects. The artifacts are all associated with every cell for the most part, right? So you can compare cells within that. If you start comparing different samples, and you treat cells as independent events, no, that doesn't work at all, because those cells aren't independent. They're sample dependent. If you compare 10 samples, all 10 samples have different, characteristics in their cells. That you can't just say there's 10, all these are all just cells we're comparing. It does not work that way. That gives you this pseudo replication. It massively inflates the significance. It gives you a lot of results that look good and are publishable, but they're not correct. And papers took a long time to catch up to that. You have a lot of papers that are out there that aren't real. So this is a very important concept where you need to think of, and it's really easy to solve this problem. You just do pseudobulk. So if you have different samples, you just take the clusters in different samples, make them into pseudobulk, right? So each cluster has an averaged expression, right? And you compare these suitable averaged expressions across samples, right? And that's the way you can get around this significance issue. So, and one thing to know about that with cross samples comparisons is you need one. So basically, if you're just comparing, like, I want to compare these 3 samples or these 2 samples, you can't just compare 2 samples. There's no significance in that. You have to have at least n equals 3 just to compare samples. Just I'm comparing entire samples, right? So we need, let's say I have 3 to 5 samples I need to compare. I want to compare the differential genes across all the clusters. I can make a pseudo bulk of each one of those samples, and I can compare across, you know what I mean? And I can also make pseudo bulks of those cell type clusters. So each cell type has its own pseudo bulk, and I compare across those samples. That's possible. If I want to look at things like treated versus untreated, so there's a condition associated with each sample. If I want to do that, I have to have at least n equals 2 for the condition. So if I have 2 samples and they're both treated versus untreated, I cannot do that's not good enough. I need n equals 1, n equals 2 per sample per patient. So I would need 2 patients per condition. Does that make sense? So I need 2 patients that, you know, I need more than one. So I need 2 patients with 3 patients basically with each one condition, right? So you need each sample has to have 1 replicate, and there's one has to be 1 sample per condition, right? Per condition. So the point is, if you had one sample and it was treated versus untreated, that would not work, because there's only n versus 1 per sample, right? There's 1 treated, one not treated, right? So we need 2 patients. If I had 2 patients, and for each of those patients there was a before and after, treated versus untreated, then that would work. You see what I mean? So you need to have n equals 2 per the condition, right? So if you have 2 patients, then you have 2 conditions, right? Or 2, n equals 2 per condition. But if you just want to compare samples, then equals 3 is fine. Okay, that makes sense. I went around in a circle there because I started getting mixed up between n's and samples, but I think that makes sense. You just have to have enough replicates to be able to do it, and you can't make up replicates by pseudo replicates by just saying cells are independent. So if you call back to workshop 1, this is very simple to using DSeq2 on bulk aggregates, right? You create these aggregates and then you run DSeq2 on them. Not hard at all, right? So in our sample, we only have 1 sample, right? So we can't really show you how to do this without pulling in more samples, and even then we wouldn't have enough per condition. We have to pull in 3 samples to get going, and we're just going to show you how to do it hand wavy, and you can apply it later. So within our own sample ourselves, we can do aggregate expression, group by cell type with our RNA-si. And what that's going to do is it's going to aggregate a pseudo bulk signal for each cluster. And that would be great if you wanted to. And we could also compare the clusters now at DSeq2 if you wanted to. But what I would show you in real life, what I'm talking about, is you would do group by here. You put your sample ID and your cell type. So in that way, for each sample ID, you would have for a pseudo bulks for each cell type, right? Then you could compare back the cell types among each of the samples, and you would do it, and that's how you would be able to do that in the correct way. So hope that makes sense. Sorry to go around in a circle there. And you can see here all you would have is like an RNA-seq matrix at the end, bulk RNA-seq looking, right? Your cells, and for each cell type you have your counts. So it's just really averaging the counts together to where it makes sense to be able to do. All right. So sorry I went a little crazy there, but I think that makes sense, and we'll show you an example later in practice problem 1. So let's just kind of move on now with our gene set analysis. We started out with our marker genes and found our differential expression between different clusters, and now we're going to move the next step and do our pathway and gene set analysis. So first we're going to use FGCEA just like before, really, honestly, to do our gene set enrichment analysis per cell type now. And it's very similar to the same workflow. We pull in our hallmark sets, and then we pick 1 tumor cell type. We could have used the genes. We could have just said tumor against T cells, the ones we just did before. But let's grab, just to show you the principle, let's grab tumor epithelial against everything. So this is genes that are expressed just in this cluster versus everyone else. So it's just the major marker genes for that tumor. We grab these and we put those, we rank them, and then throw them into FGCA just exactly like workshop 1. and we get a list of pathways here that are have a positive or lower NES. The higher the NES, the more associated with that the differential genes with that cell type, right? And what we see is over here in the tumor, we have estrogen that's estrogen pathways and KRAS signaling down, and over here on the right, it's depleted in the tumor, which is kind of everything else is all a lot of kind of immune related things that are showing kind of immuno activation. And that kind of actually makes sense because the depleted pathways are exactly what we'd expect. They're all depleted in tumor cells, right? So that makes sense. They should go toward the immune. On the right here, the estrogen is actually not crazy. Honestly, every time I do an analysis, I find for differential expression, I always find estrogen. And what happens is there's shared epithelial transcriptal programs. There's nothing to do with gynecological epithelial cancers. They just reflect shared transcriptional programs of epithelial cells. And so it's like a learning thing anyway, because you'll see this a lot, I guarantee, among different cancers in your analysis. And just know that that's not ridiculous. That actually kind of makes sense. And KRAS signaling down actually does make sense on the tumor side. So the pathways seem to make sense, and they are going toward what we would expect. And then let's move toward the GSVA, or per cell pathway. So that's when we want to find the score per each individual cell for the pathway activities. That should be pretty cool. So we'll go ahead and grab our SA. This time we use SCT, remember, because it's a visualization. We're going to look at our pathways. We want to look at the score, make a list of those, and then run our GSVA just like before. We just have our expression matrix and the gene sets we're looking for. The only difference here is SSGSCA for single cell. That gets us going a little bit better for, you could use GSVA, but this is better than single cell version. And we go ahead and get those pathway features, and instead of looking at the clustering, we're going to project them back onto the actual spatial plot and see what's going on with that. So for our EMT, we see kind of a diffuse myeloid signal. Interesting. Hypoxia up here in the monocytes again, but really diffuse. Inflammatory, even more associated with monocytes, but still kind of diffuse. Interferon gamma on here, and on the T cells and the monocytes. ETF targets really stuck in here with these cycling cells. ETF normally you expect to see them in epithelial because you have a lot of proliferating cells in tumors. But since the tumor population is so small, that's probably why we're seeing them up here. And we have OXPHOS up here. Interesting. Once again, in the macrophages. Macrophage is really doing a lot in this sample, right? We also see a lot in the cycling. So what we're seeing here, we saw EMT is elevated in the myeloids, which represent that mesenchymal-like plasticity, right? We have hypoxia or broadly elevated across all the cell types. It's kind of common to see in these widely expressed metabolic genes. Inflammatory response is myeloid dominant. Interferon gamma response is in the myeloid and T cell compartments. This is kind of consistent with what we're seeing in activated, TME. We have the ETF targets, like we mentioned, and the T cell, cycling T cells, and OXPHOS is highest in the myeloid. Things we expect to see. And this is what bulk RNA-seq can show. Pathway activity varies by cell type. And the patterns don't always match expectations. And you can't uncover that with bulk RNA-seq. And this is the beauty of single-cell RNA-seq, that we can see these underlying patterns, right? And then we can go further and look at, we can do our just Venn plot of our expression to kind of see if things end up where we'd expect. And, and what we're seeing here is just kind of more continuous, not binary, enriched, non-enriched. And we're seeing things like EMT and endothelial and macrophoid and monocytes. We're seeing, hypoxia broadly across, inflammatory peaks into dendritic and monocytes, all things we'd expect to see in the tumor. So our analysis is working good, and things are coming out, relatively, as we would expect. So you can feel pretty good about how, all of that is working pretty well, I believe. Okay, so now let's move on to NMF. So this is going to be working on really trying to discover our gene programs, right? So in workshop 1, if you remember, NMF decomposed the sample times gene matrix into these interpretable transcriptal programs, right? Well, in single cell, we're going to kind of move that a little bit further and to apply the same logic to really a cell times gene matrix, right? Where the programs can correspond to actual biological states rather than just patient subtypes. So let's see that. So to do this, for NMF, just like before, first we're just going to grab, we're going to grab our tumor cells. This is what we kind of want to look at. So just remember the subset object by our tumor epithelial cells. And then we'll get our variable features. We're just using Surratt here. And then we'll get our assay, basically based on those features. This is just straight up Surratt processing. And then we'll use the variable features of this for our NFF, right? So we're basically just trying to recalculate our variable features and send those over to the NMF. And then we want to do here, we're using Brunette for which requires a non-negative matrix. And so we need to make that just basically say, if you're below 0, you're actually 0. And that's okay in standard practice in NMF. And then with always NMF, we need to choose our optimal K. What is our K value going to be for this? To pick our how many cell states we need. This is extremely important. And what we're going to do is run our program like we've done before, where we had a kind of our custom little algo or our code to run the program for 50 runs each of random initializations per K between K2, 3, 4, 5, 6, 7, 8. And we'll go and figure those out. And we'll go and run those, plot them, and see what we should do best. And basically, we just ran this thing, a bunch of times, and got the results and plotted them up here based on our silhouette score that we calculated. And we'll see our silhouette score, kind of a little curve here. It looks kind of like those PCA plots. And basically, the higher silhouette usually is programs are more reproducible across runs. So the clusters are more stable, as you want cluster stability, right? As we move farther. So how we choose this, based on the paper for NMF, they will choose basically you really want a high silhouette width, but you want to basically, it's kind of the elbow, right? You want to find the last drop, right? And then it levels off. So if we did that, it's kind of that elbow again, we would choose 4. Now let's say I chose 4 and I ran it, and what happened was I found 4 programs, and it was really obvious that one program was being split into 2. And this is one of our most powerful, important programs. So I ended up changing back to 3 because and I found that retained that cluster. So sometimes, even though this says one way, you have to try and do it yourself to see what the best way is. So here, I'm going to go ahead and run this with 3 and really look at what I use, this RCPPML. This is extremely important. If you run NMF with single cell data, it's going to take forever. You don't even want to think about how long it's going to take. This RCPPML makes it run really fast. It's pretty sweet. So you just run that. K equals 3, put a C in there. It'll go ahead and run through really fast. We want to grab our W and H matrices, the split out for the NMF, and then we're going to go ahead and look at our W matrix and plot our top 15 genes per program here. And what we'll see here is the program 1 is very textbook AT2 LUAD. We have the surrogate proteins, and in NAPSA, a LUAD marker, and ribosomal translation machinery, all the things we would see in a core AT2 program. So we feel pretty good about this. This is the one that was split and makes a lot more biological sense as 1. The second one has a ton of mitochondrial genes. So the first thought is we need to probably maybe possibly get rid of mitochondrial genes to do this. But we look back at our genes, we also see things like Jun, EGR1, plus these long coding RNAs, and these are all associated with metabolic stress, where these cells are likely reflecting cells under energetic or hypoxic pressure. So they could be, that happens when people are dying too, though, right? So it's kind of one of those things where we have to look into this further. But I think it's important not to jump in and say we should immediately get rid of the mitochondrial genes, because we have very low mitochondrial expression in this sample. That is not a huge contaminating factor. So it's very possible this is a real program, and it's more stress related. So it's something to keep in mind instead of just throwing them all out. And our third one is dominated by ribosomal and translational machinery, along with S100A10, TMSBX4, TMSB10. So along with having all these ribosomal genes, once again, that you would think maybe you should throw out, we also have these cytoskeleton actin dynamic genes, right? Associated with cell motility. So this could suggest that translationally active mesenchymal adjacent subpopulation, this is a very real population. It could be real. So you don't necessarily want to throw out, if you throw out these genes, you may lose that program. So we have 3 interesting programs: one cell stress related, one cytoskeleton related, and one AT2 related. Let's go ahead and plot those back on the tumor and see what we see here. Well, the second one, the stress related one's pretty spread out, diffuse along all the cells. That kind of makes sense, right, for a stress related program. And then the AT2 is just kind of widely distributed among all the cells, which kind of makes sense because that's what it is, an AT2 Lewis tumor. And then what really interesting to me is that little tiny population. Look at it. It's really bright by itself. I think this is a real population, this program 3, right? I think we have a real like cytoskeleton mesenchymal thing going on here. Very interesting, and that would be something that really I think we should look into later to see what's going on there. It could be break down the cytoskeleton for it to move out into a EMT situation. So yeah, really cool finding there. I think that could be looked into further. All right, so now that we've kind of looked at our pathways and RNMF and that kind of stuff, let's talk about cell states and function scoring. And first I want to go back just real quick and show you talk about the T cell subclustering from Workshop 1, or really just reintroduce that. Remember when we took the T cell cluster and then further broke it down with new markers and re-annotated it? Well, I'm just going to re-show you that and kind of show you what we see here. And to do that, we kind of shifted. It wasn't so much that these are T cells. It was what functional states are these T cells? So now we know the actual what they are. Once again, what are they doing? What functional states? What programs are associated? And this is where add module score comes in. It's a really strong, powerful general purpose tool. This is where I want to kind of introduce this a little bit more. We used this last workshop to score our clusters. When we found we found the highest score in each one of these clusters to be able to justify the naming. So we're going to take add module score a lot further, and I'm going to show you what it can do. It's really cool, especially in single cell RNA. Spatial is great too, but it's a little less powerful there. There's a lot more confounding things, but it's really great in single cell RNA, I think. It's just kind of a reminder. Remember, we have our naive CD4s over here, our Tregs here, that cycling population is here, just to kind of let you know. We have our exhausted CD8s, our cytotoxic CD8s, and kind of the mixture of CD4, CD8s. These are kind of I feel like these are kind of cells that are kind of activated, but they didn't quite make it all the way to one of their program states there. So we're going to learn a lot more about what's going on here right now. All right, so let's look at cell functional state scoring. So what you do with add module score, and the cool thing about it is it scores any custom signature you want. You give it a list of genes, and it scores that into a signature. And that's pretty cool if you think about it, because a list of genes can mean a lot of different things. You could really do a lot of calculations based on genes. Even mathematical calculations based on distributions of genes and make scores. There's all sorts of things you can do with scores. But a very just the simplistic way of looking at it, we're just saying these are the genes we normally see with exhaustion, cytotoxicity, and these are the ones we normally see with proliferation. So we have 3 signatures of T cell functional states that we're going to score for every single cell type. So every cell type is going to have a score for each one of these guys. It's going to be put into a column into our metadata. So basically we just run add module score, our object, features is just going to be our metadata column we're looking at, and we're going to call it exhaustion score, and we're going to score those and then plot those back and see what we see in that T cell compartment. What jumps out, proliferation in the cycling cells, the cytotoxicity in the cytotoxic CD8 cells, and there's kind of a little exhaustion score that kind of goes over here, but also is kind of over here in the Tregs and over here in the mixtures. It's kind of bleeding over there, which kind of makes sense. So this all looks really solid. It makes sense. If you look at the violin plots, the exhausted CD8 plus cytotoxic are seeing the highest. The cytotoxicity is the highest there. The exhausted are showing the highest exhaustion, and proliferation is kind of spread out relatively evenly across, because it's like a mixture of all those cells together in that proliferation deal that we're seeing. And these are largely well behaved. They make a lot of sense, and they're like we would expect. You can read into it a little bit more. So let's look a bit further. One other comment I want to make about T cell exhaustion we can look at. Here is a very clinically important plot here. It's really simple. It's just cytotoxicity module scores, or module score, right, by the exhaustion module score. And you'll see here we have ones that are cytotoxic and exhausted. Here, cytotoxic but not exhausted. And here, exhausted but not cytotoxic. And why is this important? Well, cells with high exhaustion, right, high exhaustion, that retain their cytotoxic potential. This often predicts immunotherapy response. And they respond best to PD-1 blockade, where terminally exhausted cells cannot, right? So we kind of want this mixture, right? So like, if you see, like, if you're exhaustion only, you're not good for therapy. If you're cytotoxic only, you're a little bit better, but you're not great for therapy. If you're both, a mixture of both, these are the ones that are the cells that are most likely for the immunotherapies to work on. So this is a really important plot, I think, to show like how likely, if you look at the different distributions of cells, you might get an idea, or you might even get a numerical, a number, a hazard score, or something like that, showing, how likely, what's the survival based on your different distributions. And you can score patients and be like, this patient has this percentage of this ratio. They're more likely to be put in this immunotherapy group or a different treatment, and this can actually predict their survival. So this is a very cool plot. It's something to really think about going forward when you look at immune signals, things like that. So let's take this whole analysis way we're doing things, and let's push this over to the macrophage. Macrophages are really cool to look at in LUAD, right? Because their functional states are so important. Because you have this polarization state, right? This M1 versus M2. Now remember, there's two tumor-associated macrophages are one of the most abundant immune cells in LUAD, right? So we're going to see them here. They're going to matter. But what their polarization state is at the certain time will reflect what the tumor is doing, right? So what does this mean? So we have two major states, and I don't want to dumb this down. M1, M2 is a very dumbed down version to look at this, and macrophage people will get mad at me and be like, you can't say that. There's lots of different kinds of M1 and different kinds of M2, and there's a gradient and all this stuff, and that is true. So we're just really oversimplifying this, and we'll explain how that is a gradient. But just the overall top-level look at macrophages, M1 are pro-inflammatory anti-tumor. Now, what does this mean? Let's say you have a cut on your arm, right? So that's a problem, right? There's a lot of things going on. We need help. The body needs help. We need to react to this. So the M1s come first. They have a pro-inflammatory response. All of our immune cells come there. Everything comes to fight any bacteria, any problems, help to heal, or really just help to start the clotting, get things started, like just all the different things that you would go to have a pro-inflammatory environment to react to damage, whether it be a cut or a tumor or whatever, right? And this is like anti-everything. However, once you've kind of dealt with that a little bit, there needs to be healing and growth, right? So M2 comes in and knocks down that immune response. If you don't get rid of the immune response, you can't ever heal. So it knocks down the immune response, and it creates a pro-growth environment, which you could say is pro-growth tumor or pro-growth whatever, right? But it's really pro-growth, which by proxy is pro-tumor, right? So it knocks down the immune system and provides, vascular machinery to come in. It wants to help heal and grow that cut, right? Or heal that problem it sees. That's not good though, right? If we get to M2, then the tumor will have, it'll be very immunosuppressive, and the tumor can do bad. Conversely, that could also be bad for immunotherapy because you don't have an immune response to treat, right, with immunotherapy drugs. So one reason why immune cold tumors are so dangerous. And immune cold plus M2, really, really bad. Anyway, so let's talk about this. But really, it's like the next step is to say it's not really polarization. It's a spectrum or a continuum, right? Macrophages really always express both programs at the same time. It's just how much. Is it 99% one, 50-50, 70-30 of which one? That's what you need to learn. That's what's important. Okay, so let's subset out just our macrophage monocyte clusters, just those two clusters, and rerun all of our SERAT pipeline and recluster it just like the T cells. And we see here we have 5 different clusters myeloid. We're not gonna take the next step of actually naming those right now, but we'll kind of, as we go through it, it'll be really patently obvious what's going on here, and this is what we need to work on here. No point in going through all that again right now. You guys already know how to do that. So first, let's just look at the clusters, and one thing we can kind of see here, one thing you can do is we see a tiny cluster. Well, maybe that should just be one. Like, maybe, is that cluster really different? So we're just going to jump in and look at that really close and find all the markers for everybody and look at the top 5 markers per cluster, grab the top 5 from 5 and be like, okay, top 10 from 5, and be like, okay, are these real markers? Do they make sense? So we just taken that cluster, found all the differential expression genes in that cluster, just building upon what we just did earlier, right? And then we're just looking at them and seeing what's up. Do they make sense? So this cluster makes sense. And what we're seeing is, yeah, it does. FAB-Pu4 is a canonical marker of lipid-associated macrophages. It's well characterized subtype, and the TMA, GPD1, and let's see, PHLDA3, reinforce like a lipid metabolic program, INHHB1 associated with immunosuppressive macrophage polarization, and so on and so on. They seem to actually make sense, and we can say, yeah, this probably is a real cluster. So let's just leave it as, just wanted to show you how you can kind of, just get you to think about how you deal with these situations when they arise. So it won't just be obvious. Because what if that wasn't a cluster? Then we'd have to recluster, do a lower resolution, or combine clusters, things like that. You can always just combine clusters. So let's go ahead and do our add module score and see what's going on with this new myeloid population. So we're going to make our new signature, our own classic signature, and then we're going to score that. So first we're just going to look at it on a dot plot just to kind of show what the signature might look like. And we see the different clusters, and we see the different genes that are associated with them. You can see cluster 5 really jumps out as being different. So yeah, there's similarities and differences between these two. This is what we want to expect in some kind of a continuum. And you can see things like cluster 3 here. It's a very classic monocyte population with S100A8, S100A9, FECAN, FCN1. So this makes sense. Polyinflammatory monocyte. Clusters 1 and 2 look like more M2 things with high CD163, which is a great marker for M2. MRC1, FLR2, these are all things to expect to see in M2. Where cluster 4 kind of shares M2 markers, but also kind of a mixture. So that could be kind of a gradient. So this is kind of a little bit what we could see, looking at, we kind of, a little bit idea of what the markers are pointing to. Let's go ahead and score these. So we're just going to take, so this is scored just for the entire panel. This is going to score for specifically M1, M2. So we're going to look at our M1 genes, M2 genes, score those with add module score, and replot those and see what we get. And it's a very great plot. Very great plot. I mean, we can really see the difference here. You can see it's very anti-complementary. M1 signal is largely diffuse across this lowered environment, and with the strongest focal activity up here in the top left, where M2 is just very strong here. So it's very clear in the bottom right here. And I'm concentrating the lower cluster 2 and on the right side cluster 5, and you can really see the difference here, right? And this is really consistent with that CD163 MRC1, and we're seeing this M1 region is the most blue and most red, so that makes sense. Everything's looking really good, and we're really seeing this split of what's going on. On the top here, we're seeing this pro-inflammation, and here we're seeing pro-tumor. And so this is very interesting to what's going on in this myeloid compartment. And remember, this is not a spatial. It's hard to look at this and not think of spatial. These aren't separate from the tumor. They are inside the tumor around the microenvironment doing shit. Doing stuff. I got too excited about science. They're doing stuff, right? They're intermixed around areas, and that's why spatial is so cool too, right? But don't think of them as by themselves. They're doing what they're doing out there, and it looks like we have more of an M2 population in general in a more immunosuppressive environment, probably in this tumor. And let's kind of look at another plot, kind of like before, and we see these are the dominant M1s, dominant M2s, and the hybrids. And we'll see we don't see many hybrids. It's mainly M1, M2 with some bleed over, right? And mainly M2 if you look a little bit closer. We won't go too much into this, but it just kind of it just supports what we've been talking about earlier about these clusters kind of matching up the way we'd expect. And you can see here, we'll just kind of subtract M2 minus M1, and we'll see here, it kind of gives you a measure of how M2 they are, right? These are the most M2, these are the most M1. You can see things like, cluster 3 here is the most pro-inflammatory, right? The classical monocyte cluster. Cluster 4, it kind of leads M1, but it's still kind of in the middle, right? It's kind of that transitional inflammatory TAM, right? Where we go in the pro-tumor. Cluster 2 is the most skewed, right? With a CD163, MRC1, full R2 probably, or not probably, it has it. And then we see these kind of lesser ones, and we see this gradient. This is a transitional populations between them. So I think that worked out really good. You can see how we can take a population, recluster it, and use add module score based on specific signatures, right, to find gradients and continuums inside our populations. You can kind of look at the distribution of cells if you want to, again here, and you'll see things like the highest M1 dominant is that cluster 3, the far left, right? And the highest M2 is the bottom right, that 2, right? So you can kind of see the distribution. You can also see the ones that are kind of in the middle here, like 4. They're probably really interesting, right? And you can also look through the genes as we had before and kind of look and see, do these, M2 guys, are they in the right populations, things like that. And we kind of see here some of these M2 ones that we saw that were the highest in certain areas that we thought were important are also kind of expressed across everybody. So that kind of gives you that continuum of M2 dominating across everything. And it's always important to look at these two because they give you more insight into what's going on here, but everything kind of makes sense with a little bit of a couple of catches, and I'll give you guys a chance if you want to go through those a little bit more, a little farther. It might tell you a little bit more, in depth about what's going on biologically, but we won't really. We've kind of already kind of covered that. You see what's going on and how what you could find, right? So what can we do behind just looking at cell types? Because cell types are actually not the best thing to look at. What we showed, cell states, continuums, gradients, things like that, right? Biological programs, pathways, lots of things, right? This is the cool thing about module score. It can be anything. Your own specific signature. It could be a translational signature. It could be, we'll talk about later. It could be a public signature, for treatment, anything, right? There's lots you can do. So let's just talk about how you could do a pathway, just really quick, just to show you an example. We loaded our pathways back. It's going to be our hallmark pathways. We scored each of those. So for each cell, there's a column for each hallmark pathway, and it gives it its score. And then we'll just throw those scores back over and look at them on our actual tumor compartment. What do we see? Glycolysis in the cycling. We see, hypoxia kind of spread out like we saw before. Interferon over here. OXPHOS up in the cycling. Things that we expect. It's not great, but it's not perfect. It's not perfect. It's not bad. It's not as. I think it's kind of the results kind of mixed here. Some are good, some are bad. I think this IL-2 stats one is really good. It's something to kind of focus on, I think. You can see here, a lot of these add module scores didn't really work that well. They didn't jump out a lot. And that doesn't mean they're wrong. It just means instead of hallmark pathways, maybe you should try Keck pathways. Maybe you should try different hallmark pathways. Who knows? You can try different things. They're not all going to work. That's the whole point of science, right? We got to try things. But what we do see, let's just kind of focus on this IL-2 stats because it's kind of interesting here. I think this signal here. And you can see that this Treg here shows the highest median and the only meaningful positive tail of this with CD4 plus helper cells positive and every CD8 subtype hovering at just below 0. So this is textbook biology, right? You have the IDL2 STAT5 signaling underwrites this Treg identity, right? Which really is based on FOXP3 maintenance and CD4 helper survival, while these CD4 effectors tend to rely less on this IDL2, right, once they're actually differentiated. And the fact this clean pattern emerges from this panel is very reassuring, and it tells you the scoring is significant enough to find real biology. However, you have to have the right signal to look for, right? It's not going to find anything, and this was found well because it exists. The others didn't find things because those things don't exist, interestingly, in this panel. So no one's wrong. It's just what we're seeing, right? And there's different ways of looking at things, and sometimes GSVA is better, sometimes add module score is better. But the bigger picture is you can use this as a universal scoring tool, right? And basically what it's doing, just to kind of tell you, is it's looking at the expression, it's looking at your genes, It's saying how much of these genes are expressed compared to some control genes. So that's all it's doing, and it's just saying, hey, compared to control genes, these expressions are a little bit higher of these genes in these cells. And then, like I mentioned, there's just all you can do with this. But the workflow that really holds across every application is you can curate the gene set deliberately, right? Your own custom signature, but always check for coverage before, right? Because you may need to know like how many genes do you need to be there. Make sure they're there actually. You may not have enough, right? Always start with a seed, and then try different resolutions for your spatial structure and validate against individual genes at the end, right? But man, it's a really good way of doing things, and a lot of cool things you can do with it. Okay, so that being said, let's move on to the cool standard analysis you normally see in single-cell RNA-seq, and that is trajectory analysis and cell-cell communication. All right, so what does this even mean? So trajectory analysis is an approach where you order cells along a pseudotime continuum. And by doing this, you can reconstruct developmental or activation paths from basically a snapshot of cells captured at different stages. One important thing is not every data set has a trajectory that's meaningful, right? It works best when cells represent a continuum, right? T cell activation to exhaustion, monocyte to macrophage differentiation, epithelial to differentiated. When you have this kind of continuum, it's the best place to find these. And if you don't do that, it ends up doing things just like clustering. Instead of finding like a continuum, it'll just find two clusters, and it's just kind of meaningless to do that, right? So you don't always run this. You just run it when there is something that you feel like you can see in there. And since in cancer there's often immune situations, you most often have something you can look at, and we will show how that works. And so what is actually happening on this trajectory? How is this working? So this is, I will say, highly dependent on your clustering, right? Your clusters have to be right, or this absolutely will fail. So, because what is happening is, we're going to use Slingshot. What Slingshot does is it looks at each cluster. It finds the centroid of that cluster, right? Then it makes a minimum spanning tree across your entire plot, your UMAP plot, right? And it connects the centroids of all the different clusters. Okay, and that kind of gives it a tree map, a map of all the possible trajectories that could happen, right? And then it basically runs an algorithm that cuts up the trees and clusters, different branches, and finds the most meaningful paths that actually makes sense to path through the data. And then it does a smoothing function basically because it's forcing it through the centroids first, so it wants to smooth that so they'll go through the right distribution of cells in each cluster. So that's kind of how you're finding your trajectory. And then you can look at things like the differential expression. You can basically project genes along the path of that pseudotime, because you can basically calculate this pseudotime. It's just the distance from the root basically to the end. And you project these genes along this route, right? And try to understand the differences between the different genes and different lineages. That's basically what trajectory analysis is. And long story short, it needs to be baby. One thing, if I could teach you one thing, things like inverse CNV, things like copycat, things like trajectory, cell chat, basically any single deep learning method you're going to see online, they all need to be babied. None of them work out of the box. Whenever you look at their paper, they always work out of the box, but that's never the case in real life. You have to baby these things. And what I mean by baby is you already have to have a pretty substantial understanding of the biology to be able to point them in the right direction and understand if they make the wrong result. And I'll show you what I mean in a second. So for Slingshot, TreadSeq, so I'm using Slingshot here, and the reason I'm using Slingshot, there's other methods like Monocle, for example. Monocle 3 is very useful, but a lot of the other methods are very complicated installs, and I wanted to keep things kind of simple for you. And Slingshot still works pretty well and is widely cited as well. And TreadSeq is a way of doing your differential expression on your trajectory. So this is really still gold standard way of doing things. You don't have to worry about it being something from the past. So first we need to do single cell experiment instead of Seurat. So we just take it and flip it to a single cell object and basically run here. And you can see here we run Slingshot always set seed based on our clusters and our UMAP. Remember, we're going inside of our T cell, by the way. We're doing our minimum spanning tree in our T cell population, just to kind of show you how that's working. And so make sure your clusters are the right ones. And we're going to choose UMAP, and then our start is going to be number 1, and our ends are 5, 2, 6. And what does this even mean? Okay, so if you run this without these columns, it will find them themselves, right? It will find a root somewhere that feels like it makes sense, and it will make a tree in any direction it feels like it makes sense. And it's going to be wrong probably almost every time. And when I tried that, it automatically went to the most differentiated and went backwards to the naive, which makes no sense. Kind of like copycat. I like to do things just the opposite way. So I realized I need to baby it. I babied it a little bit. I just said, hey, this is where you need to start. And I basically said, I validated with CCR correlation that the top right cluster is a naive CD4 plus cells, which remember that top right one was. Remember it was CD4, CD8 on the bottom left. So we were able to show that one was the naive population. That's where it should start. So I set the root to that. I ran it. I left this out. Still got it wrong. So then I was like, okay, it's almost like the copycat. I'll have to just tell you everything. So I'm telling you everything. I'm saying start at 1 and go to 5, 2, and 6. Those are your endpoints. And whatever happens between those, that's your job to find the path through. I'm not going to give you everything. And these all represented states that made sense, going from naive to Treg to mixed to T helper to static sockets, all things that kind of made sense, looking to other clusters they could go to. Okay, so I basically just ran that. I already ran it. I kind of just expected the structure of this thing to show you what it looked like. The sling lineages will show you what it looks like. And we see here that, 1, 5, 1, 2, 1, 6, but what's in between? So how to read this? I'm just going through lineages here. And these are just the clusters we have, we've already made earlier when we did this subset of the T cell compartment. So how can we do this here? So lineage 1 starts out at naive, CD4 plus, it goes to exhausted helper, then it goes to proliferating, then it goes to cytotoxic exhausted, and then it goes to pure cytotoxic. Kind of makes sense. This one, look, 1, 4, 7, 3 are the same. Just the last step. This went to pure cytotoxic. This took a weird turn to a mixed population. So that'll be interesting to look at. And our third lineage goes to 1, 4, 6, which actually goes to exhausted helper cells and then Treg, the CD4 Treg arm. And this is like the most shortest, most direct one of these lineages. And all of these, paths make biological sense through the data. So these are all real paths that actually make sense, and their scores look really good. And we actually plot these back on to our UMAP plot of our T cells, and we'll ignore the weird one, and we see the Treg one just kind of cruises here to the bottom through helper to the Treg ends. This other one, moves through the mix, through the cycling, through the partly cytotoxic to the pure cytotoxic. This one goes through here, through cycling to the green, but then instead of going blue, it curves back to this mixed population, CD4, CD8 mixed. And where this represents pure cytotoxic cells, and this represents more of this like activated cells that aren't all the way to the end of their lineage, that they didn't move to the end of their continuum, right? And what we're seeing here is, I'm trying to remember what to say. So what is going on with this loop? So these all make biological sense the way they're going, but there's this weird loop happening here. Why is this loop happening? So what's happening is this is the common artifact of the math and trajectory analysis. And because of this stupid, silly cycling weird thing, and we could have removed this. Remember, a long, long time ago, we could have removed this population. It's been a thorn in our hide the whole way, but it's a cycling T-cell population. It's real, and I don't want to remove it. So you could have removed this, and this loop would have gone away. It would have gone right here and ended, but I don't want to do that. It's not the right way to do things. So we could either just remove that trajectory completely for the final publication plot, or leave it as is. I'd probably just take the top one off, and then throw it in the main, and throw this in supplementary. But there's nothing wrong with this. It loops back because of some complicated mathematical artifacts. It doesn't mean to loop back here. It's just doing its thing. It stops. You can just put a line here and stop it. So that kind of explains how that is going to work. So because that one's kind of weird, we're going to take the 1 and 3 and take a look at those, take it a little further. So one thing we look at first, we want to make sure, are these going in the right directions? Because they were opposite before. And we checked the CCLR versus pseudotime correlation of these lineages. They're all negative. That means we're going in the right direction, from the naive to the cytotoxic. It makes sense. Now, if these, you can read here and kind of see if they were opposite, we'd have to run this thing to kind of flip it. And that can't happen. You always got to watch for those. Make sure your lineages are going in the right direction. And we have several different means of validating that going forward, so we feel good about that. So let's go ahead and just plot our pseudotime. Take the two simple ones and plot them. You can kind of look at the clusters here again. You can kind of remember our T-cell clusters. And you can see here, lineage 6, it kind of cruises down here, right in the difference goes through these three clusters. And the right one, it goes through down here, which is down here, back to the cycling, right? And through here. So it just goes through these. It skips that one, kind of cruises through there. And it makes a lot of sense. This result here, it originates in the naive CD4 plus T cells in the upper right. It flows cleanly through the CD8 compartment, to the cycling, the CD8, and it stops on the most cytotoxic, where 3 ends up, going down to the mixed population and to this Treg population, the terminal CD4 state. But everything kind of moves through really well, and it looks pretty good. So I feel pretty good about our results. The next thing you would do is kind of look at plotting genes along this pseudotime, right? So what genes can we plot along the pseudotime and see what's going on? So we went ahead and grabbed some genes just to see what's happening and just plotted them along the time, like what is their expression along the pseudotime, and just to kind of see what's up. And we see some things that kind of make sense. The CCLR7 peaks very early before dropping to 0, which makes sense because this naive state, where GZM rises at the top, which is more a cytotoxic duration state, and so on and so forth. The more naive ones start at the top, other ones go at the bottom. This one's kind of interesting. It goes up and down. So what does PDC1 mean? It means there's a modest, it kind of plateaus then persists with a modest exhaustion still present. We're just kind of looking at what's happening between the different states, right? So that's the way to do it, kind of hand wavy. We're just looking at it, right? But we need to know this like with actual statistical certainty, right? Significance. So that's what TradeSeq can do. It can run this negative binomial GAN for each gene along the trajectory and then perform statistical tests to identify genes that change significantly over pseudotime. So do genes change along the pseudotime, and do they change between the lineages, and do they change between the endpoints of the lineages? That is what we're going to do. So first we just grab the variable features, a basic assay, and we fit this GAN, which just uses our slingshot object, our counts matrix, and just runs that GAN for us. And then we have that result. We can run this association test and this pattern test and this differential test. Those are the 3 things that you can do with TradeSeq. And what you'll see here is, first we'll do the association test, which is saying which genes change significantly along any lineages, right? This is the most powerful general test. So we run that, and we see here all these genes. So all these genes among any lineages, doesn't matter. These are the ones that we go along the tumor time, we go along the pseudotime. These are the ones that are showing differential expression, basically from the end to the front, really, as they move along, right? And we can see here, we're looking at more statistical versions of how these move along, and see if they match our pseudotime. And you can kind of look at a few, like the FOS. Where's FOS? It's an interesting one. Starts high. It's an immediate early gene marking in an activated naive state. You see things like STM1, these peaks here, right? These actually make a lot of sense because that's the proliferating cluster. So they're very proliferating genes. So as you can see, you can just kind of follow the genes along to the time and see how well they actually fit. So what about if you want to know, do genes have differential expression across lineages, and what are their endpoints? So we can do our pattern test and our differential test to do that. And we kind of see the genes here. These are the genes that are between pathways. These are the genes that are most differentially expressed between lineages, right? And we'll go ahead and plot those, and then also show you which ones are different at the end of the endpoints, right? So these are kind of just plotting our genes here, and you can see that the blue and the purple, the green is the one that goes that pure cytotoxic state. The yellow one is the Treg one, and we can see here Treg obviously is going to flip down among all of them. But what's really cool is to kind of look at the endpoints like CCL5, June. As you can see, there's obvious changes in the endpoints and the pseudotime at the end of those populations that curve off different ways, right? To be expected to see those kind of things, right? Right, so this is the way you can look at genes, how well genes, or how much genes differentiate along these pseudotime pathways, and how much they differentiate between different lineages, and how much to the different time points end. So these are all ways you can do this. And here's a way of just visualizing the time points. For instance, these are higher in lineage 1, these are higher in lineage 2. Because it makes sense which way they are going at the end. Those endpoints are different, right? The exhausted versus cytotoxic. So we can kind of see it, go through that. It all makes a lot of sense, and we feel, like reasonably good that we're doing a decent job of explaining that. All right, so now let's move on to cell-cell communication using cell chat. So it's a hot topic. People like to look at the way cells are communicating with each other, right? Like cells in the tumor microenvironment don't exist in isolation. They constantly communicate through these ligand-receptor interactions, right? For instance, a tumor cell expressing PD-L1 signals to a T cell expressing PD-L1 receptor, right? And that's how things get done. So it'd be cool if we have ability to look at these reactions to uncover how they work in our tumor sample. And we could do that using cell chat. And what cell chat does is it has this curated database of all the known LR pairs, right? And infers this communication probability from the co-expression and permeation testing, right? So it just basically, it has its own database of how the pairs already look or should look like, right? And then it just maps, it just looks to the actual cell data, looks at co-expression and does permutation testing, and maps these different receptors. And we kind of just have this basically, end up having this, having this basically probability of communications between cells. And that'll be able to see how well cells communicate toward each other and how they receive from each other, all these different things we can find out. And so really, at the end, we'll know which cell types are talking to each other through which pathways and which ligand and receptor pairs are actually driving this communication. So the cell chat workflow is pretty straightforward. We go back and choose RNA, and we just normalize it like general, not SCT, and we just kind of run through a string of kind of basic, we create the cell chat object, we pull in the cell chat database, we subset it, we identify overexpressed genes, we identify overexpressed interactions, we communicate the probability of these interactions, and then we filter that, and then we kind of put this back together and aggregate it into a way we can kind of look at it a little further, and then we can pull out these results to look at the answers. And here we're going to pull out a couple of results here on top. We just want to look at the count per source breakdown. So on the bottom left here, we're looking at the interactions that we plotted out from here. And on the right here, you see here, we're looking at the macrophage outgoing signal. So what are we actually seeing here? So this communication network, the size of the node tells you the cell type abundance. So we see a lot of T cells, macrophages, monocytes, right? And then the edge thickness tells us the communication intensity. The higher the probability of these autocrine self loops or these different strands moving on branches to other things. So the thicker it is, the more probability, more intensity going on between those two things, right? And on the right here, we have an outgoing signal. So for any, we chose macrophage here, but for any of these different nodes, you can choose to show all the outgoing and incoming to that actual node. I just wanted to show you the outcoming here. So we see the autocrine loop is very powerful. So macrophage back to macrophage, and then macrophage to monocyte is pretty powerful. Macrophage to TC is pretty powerful. Things we'd expect, but tumor epithelial is super low. It's unexpected. What we'll later see is there's a strong communication from tumor back to macrophage. So it has an incoming signal from tumor, but not outgoing. So this is very interesting to see. Just a quick look at what's going on with those deals, right? And that's how you kind of read those and kind of get a feel of what's going on. And then we could actually, for this long function here, we can actually plot out which just kind of who sends, who receives the best, right? And we see macrophage. So we see here the outgoing interaction strength is at the x-axis. The incoming interaction strength is the y-axis. So you'll see macrophages, they're going both ways, right? Where these guys are really not doing anything. And you see things like the tumor dendritic cells, they have a strong outgoing signal, but not much of an incoming, right? Where we see things like T cell, they have a strong a small outgoing and a strong incoming. So you can already kind of just get a very big overview of what different clusters and ligands and receptors are associating with each other. And then we can actually plot these out in a heat map and be like, okay, if we choose some pathways, standard pathways, how can we look at things that are basically, how can we look at outgoing and incoming signal patterns? So basically choosing the top 30 pathways in the basic shell trap network that are already in there. And then we were just running this net analysis signaling role heat map with our cell chat object. We want top pathways, just outgoing. And we get this guy. And when we're outgoing, macrophage is really strong. Tumor cell is pretty strong too, outgoing. Some other things like dendritic cells. So what's outgoing? Outgoing is dendritic monocytes, right? So monocytes, where are you? Monocytes, they're strong too. So we're seeing very strong signals across these pathways going out. And we come back in, we're going to see things more like the T cells are bigger there. Tumors kind of both ways, but left pathways. Macrophages, monocytes, they go both ways. So this is just a good way you can see the intensity between outgoing and incoming signals of every cluster along certain important pathways, with the highest ones being up here, And we see a lot of interesting stuff there. We see MHC1 is sit broadly across nearly all the cell types, but received primarily by T cells and NK cells, consistent with this widespread cytotoxic thing we've seen, where MCT2 outgoing is concentrated in really this professional antigen-presenting cells, B cells, dendritic cells, macrophages, monocytes, with incoming, a lot of just an autocrine loop, right? A lot of this self-talk going through. We see MIFs, the MIF signal sent across a lot of cell types. We see SPP1 sent strongly by macrophages and monocytes, yet they come back too, and so on and so forth. You can kind of see the different things that are going on in those different cell types. So it's just a good way of looking at this. Now, taking it a little step further, one of the most biologically interesting interactions in cancer science is this immuno-oncology interplay, right? Where how do tumor cells communicate with immune cells and so forth, back and forth. So if we understand these pathways better, we can see what's happening better with immune evasion, recruitment, suppression, things like that. So for here, we're choosing our own set of pathways, and we're doing this kind of bubble plot here. So we have our pathways here, and we have all our directions we've chosen. So we've kind of chosen our directions: tumor source, tumor target, and that's how we chose the x-axis places. And it's just saying, if we go tumor cells to these guys, what are the probabilities based on these? Or where are we seeing probabilities light up in different things? And that gives us understanding of what's going on. We see things like MCH2, CD4, the HLAs. They're all in macrophages, and tumor epithelial to macrophages. So that makes sense. TGF-beta1 is exclusively in macrophages to macrophages. This suggests this autocrine TGF-beta loop, right? SPP1, once again, macrophage. You can look at all these. You can kind of once again get a better understanding of the communication between these different cell types. How can we look at some of those interactions? I want to know just some of those specific interactions. Well, here I'll look at all the significant interactions and pick out ones that are interesting. So for each of these, source to targets, we have the ligand receptor interaction and the probability of actually happening. What jumps out is MIF at the very top has all the top three, and this APO-TREM is the top four. So looking at this, hey, let's just try. Why don't we try out, because tumor epithelial is going to T cells and macrophages here. Let's check out this MIF signal and look a bit closer to it and see what we find. And if you want to read a little bit more about it here, you can go through that. And then let's go ahead and visualize this specific pathway. So we pulled out the top pathway, which is the MIF, and we plotted out the network. And what do we see here? We see a very strong tumor. Macrophage, dendritic cell. This is really, the tumor is really going on. It really has a lot of talk here, right? Between dendritic cells, macrophages, monocytes, NK cells, T cells. Also a decent autocrine loop. So that's kind of interesting what's going on. And when we look here, the tumor has is ascending a lot of stuff here and receiving a lot. Endothelial is not receiving anything or sending anything. It's kind of interesting. So there's nothing happening in endothelial or B cells, pretty much. I guess the B cells are receiving. But what I'm seeing here is very broad. There's a lot of things that make sense. Like the sequence is very broad. Nearly every cell type pair has an edge in this plot, right? So it's just showing MIF is really a pleiotropic cytokine acting across nearly every cell type pair in the TME. And this breadth makes it interesting but therapeutic and complex, right? Because it's definitely a real signal. And Cell Chat found a real signal, but it's so broad and ubiquitous, it's really hard to see what is going on. So we need to go a step farther and be like, okay, we need to do like a ground truth check and actually seeing, does this actually make sense? Does the ligand actually appear in the source type cell? And does the receptor actually appear in the target cell to an enriched amount? If they're not, then how could it even happen in the first place, right? So that's what we're gonna do here. We're gonna find the top LR pair for this pathway. And we see here, the ligand is MIF and the receptor is CD74 and CXCR4. So let's go ahead and plot those out in the violin plot like we always do. And we're expecting to see tumor epithelium high in the MIF signal and CD34 high in the T cells, right? And B cells, that kind of stuff, immune cells. And, we do see, so I think the receptor is finding its right places, but the ligand is everywhere. It's not just associated with that only. So really this verification is only partially supportive, okay? So it reveals an important thing. The MIF ligand, the tumor epithelial, does show a clear bimodal pattern here, right? With meaningful expression up here. So it is a good contributor, except all these guys are contributors too. So that's not super great either. Although we do a better job over here. So the verdict really is the ligand is ubiquitous and the receptor is enriched, but in the wrong cell types for a clean, tumor to T cell directionality claim. So this interaction is up, and this is all these cell chats. This is the most important thing to learn here. Significant cell chat interactions don't mean they're right. They're hypothesis generating. And you can take it a little further and see if it's real. Because it's going to find things that aren't perfect. Like we saw, things are connected, but they're not specific enough. So you always take it with a grain of salt. I don't just get this data and publish it because it looks good. We had data to support it. If no one would ever looked at it, people may have never known. Anyway, so always kind of check it and know that you need to verify that. Okay, so that's our two major analyses that I think are important to go through in single cell RNA. And now we'll move on to multi-sample integration. Well, why would we want to integrate ever? Well, so far our analysis so far just comes from one patient's tumor, right? And that works great. But to make generalizable claims, we need to look across more patients, right? And more conditions. But if we do that, there's batch effects and problems, right? You can't just compare two people together. If I just put two guys together, that's going to arguably be single cell, especially in single cell RNA, as we talked about. There's going to be strong artifacts from the way they were prepared that day, the way the weather was, what time it was, the look on the person's face that day, whatever. It's gonna make it different. So we need to be able to correct for batch effects when we combine different data. So let's go ahead and show you how to do that. So what we're going to do first is just grab another one of the primary tumors from this data set and just pull it in, this lung T20, and basically just pull it in and do the exact same thing we did before. I'm just basically running the exact same pipeline of creating the object, same filtering we did before, more or less. And I did my transfer of cell type annotations, just like we did before. And then I went ahead, the next step was, after I did the notation transfer, I merged the objects. So merge object 1, object 2. So I just started up a new object, downloaded it, filtered it, transferred the cell type labels over to it, and merged them together. And we can go ahead and plot that out, and here's what we see here. And what jumps out to you right away? Yeah, exactly. What is going on here and here? It's extremely, when we plot the batch effects, which is the first thing you always do, is plot plot the batches back on, it's completely, there's a lot of separation. Specifically, let me look at this. Perfect example here. These are the same thing, but they're split by batch. I mean, same here. So this is definitely batch effects that we need to correct for. And how can we actually do this? So we're gonna do this in Seurat. You can use Harmony in Seurat, which is the easiest way to do this. And it's a very, very good way to do it in the first place. So the path to do this is take your merged object, right? And it's the merged CT object that we're using, and we're basically just splitting it by the identity. So we have to split it before we can correct the batch effects. So we're basically just splitting it into two different metadata, a column with an original identity of each one, and then we just put that the full merged. We use Harmony integration, PCA Harmony, and we use our features for the variable features we did before. And that gives us our merged Harmony. And once we did that, we have to join the layers, bring them back together. We split them. We're going to join them back together. Just, excuse me, rerun the same pipeline. UMAP, find neighbors, find clusters. Plot it back, and bam, looks great. Looks fantastic. I mean, look at these two. That one there that were split. They're actually together now. These are mixed together now really well too. This is exactly what we're going to see. When we plot the batch back on there, we're not seeing things clustered together. We're seeing intermixing. And even better, when we look at our cell labels, they're matching really well. So the biology cell types are holding up over more than one sample, which is really solid validation that we made good decisions. And we're seeing the same thing, like T cells, the kind Nick, we're seeing those three guys together. Things are all where they were. And we're seeing here is more tumor because the new one has more tumor in it. So to be expected, it's going to have more tumor in the epithelial, right? So everything worked pretty well. We feel pretty good about it, and we see things like the differences, like both tumors are T cell dominated, yet the new one has more T cells and the old one has more macrophages, right? These are clinically meaningful differences, right? The new tumor's higher T cell fraction and tumor epithelial content, so more tumors, more T cells, can indicate a more like immunologically hot tumor, right? Which gives us a more chance for immunotherapy, where the lung 9s is the opposite, right? Macrophage heavy composition compared to this M2 skewed polarization shows us more of an immunosuppressive tumor, right? That might resist PD-L1 therapy. This is very big. So we've just with our simple analysis, we've pretty much shown that one tumor is immune hot, one's cold, and here are some genes that can be associated with those two things, and this could likely push you and triage you to the right treatment or the right clinical trial. This is exactly the kind of thing we are looking for, and exactly the kind of thing that single cell RNA is able to pick up at a great clinical meaningful depth, right? And these are kind of some of the methods you could use for integration. We use the Harmony. You can also use our PCA as a common one too. And then here's kind of just putting all of the analysis together. The things we have done through the part 1, 2, where we've done what cells do we have here? We grabbed those, right? What cells are malignant? What defines populations? And then what pathways are active in those populations? What gene programs exist within tumor cells? And then what T cell states are present? And then how do cells translate between these different states as far as their trajectory? And then how do cells communicate? And then also, finally, how do we integrate things? So we've really gone over all of this. I think we've done a good job of making a lot of that make sense. And then we save everything, of course, at the end, because we don't want to have to do it all over again. And so, that being said, that's the crux of the entire workshop. I'm basically just going to take you through just kind of quickly the practice problems. I don't want to tell you everything, because I really want you to try these things out yourself, because you can learn a lot. And these aren't just waste of time practice problems that are adding on repetition, but these are actually adding on new concepts and developing things further. So it's definitely still worth doing. And so all these at the bottom are still using the same data set and that we'd used. We're going to use that merge data set, but it's all the same blue-eyed sample. And so the first thing I want to do is talk about that pseudo bulk differential expression versus cell level. Remember we talked about that? How if we want to compare things, that we talked about that when you compare gene expression between two different conditions, so two patients, treatment versus control, whatever, two conditions, the unit of replication is the sample, not the cell. If you do cell level differentiation, treats each cell as an independent observation. And like we talked about, this massively inflates the statistical significance because these cells are not independent. They shared all sorts of artifacts. This is a really, really important thing that should be obvious that people aren't listening to. So you cannot use cell level differential expression. You have to use pseudobulk when looking among samples. So now we have two samples. So let's compare the samples of this pseudobulk way and see what we can do. So first we're just going to find two clusters that are alike because we just want to compare two clusters. Cell type 1, I know it's T cells, and I know it's a good cluster to compare. We also show there's at least more than a certain amount of cells in there. And we found that it has this many cells in each sample. So we feel good about one. I know it's T cells. Let's move forward with that. And then let's pretend like, let's do it the bad way. So we basically chose our RNA, did our normalization, we joined our layers, right? And then we chose our clusters that we put together for both layers. And then here, look, we do it, instead of clusters, we do a different tumors here. So we're doing lung 1 against lung 20 here. Grouped by original identity. So we know what tumor we are, and then we're choosing our target cluster. So our target cluster is going to be the specific cluster we're looking at, right? Which is the tumor one, right? So it's going to choose, it's the one we chose here. So we chose target cluster as 1. So it's going to look at that specific cluster in each tumor and compare and we're just going to treat all the cells as independent by using finite markers and just, t-test Wilcox. And let's just see what we get from this. 20% significance. That's not good. Humongous log fold changes. So what's happening is 20% of all genes were 20% of all the transcriptome we put in there was significant. That's way more than it should. And then we see this gene, TSP1, has a log fold change of 0.65 with a 100% expression in both patients. So it's 100% expression in both patients. Yes, it's still significant. That makes no sense. So we're just getting these completely inflated values that don't make any sense at all. And look at this. This makes no sense. It's all because you're treating these things as independent in the wrong way. These are not discoveries. These are statistical artifacts of treating these cells as independent. And once again, you can do the Leighton plot, and you're seeing just way, 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 way too much significance. So how could you have done this in the right way? Okay, well, let's pretend the proper way to do this, we have to have N1 for each condition, right? So let's say we want to look at the T cell cluster. Well, we have T cell cluster, right? And then we have T cell cluster in another patient. So we have N equals 1 per condition, but we really need more than that. So it's just not enough to compare. You can't just compare 2 patients. You would have to have 1 more. If you would have had 1 more patient, then remember, we would have had a T cell, T cell, T cell. Each one condition, we would have N equals 3, that would have been fine. So when you look, we only have 2, so we'll show you how to do it, and just know that you'd have to add 1 more patient to make it statistically significant. But it doesn't mean it's wrong. You still get a good result here that can be observational, just not significant. So anyway, we're just going to do the same thing. We have our merged with our clusters, right? We've subset for our target cluster, okay? So all we have is our merged subset with our target cluster. And we are going to aggregate expression of our merged subset by group by identity. So what it's doing is it's forming it. All we have is the target cluster, right? So it's doing an aggregate expression of the T cell cluster in each one of these. And what you end up getting is just a bulk RNA-seq thing, right? So these are the T cell and T9, T cell and T20. And then all the cells have been aggregated and averaged together. It's not really an average, but it's more or less an average for all the genes. So now we have this is just a bulk RNA-seq matrix that we can do a comparison between. But we need more samples. Like, it's not enough per condition to do this. We need N equals 3 for this to move further. We have 1 condition. It's just T cell, T cell. If it was T cell versus epithelial, T cell versus epithelial, then that would be 1 per condition. That'd be okay. But it's not like that. It's just T cell versus T cell. So we need 1 more to be significant. But anyway, this is how you would do it. You would add 1 more column, and then you would just do, your differential expression analysis like you would do. You would just use DC, or whatever you want. The point is, you don't do you can only do the gene level differential expression within side of your sample. If you want to do cross patient comparisons, you have to form these aggregates of the different patients or cell types or whatever you're trying to compare to be able to, and you can do kind of that pseudo bulk differential expression. And this kind of talks a little bit about more of that n value problem, and kind of just shows you how well it worked between the two. But I think you got the idea. And second thing we're going to do, remember we did our MIF value on our cell-cell communication. It didn't work great. So we just kind of wanted to show you, let's try to build another one and take a hypothesis. And remember we mentioned that the APO trim one, so let's try to maybe we'll grab that one and take it a little bit farther. We kind of redid this, kind of showed you your bigger senders. We kind of looked at our guy again here. We're just looking at the tumor ones now alone. And then I'll let you guys look through this kind of stuff. And remember the APO trim one, we're going to choose that one instead, and get our plot here, and kind of show where a ligand should be. Like this should be in epithelial. It's not anywhere, but it is in the macrophages. So the receptor's in the right place, the ligand's not. And it just kind of failed again. You know, it's kind of a little bit frustrating. And basically, the best way to say this is, you know, let's go a little farther. So the ligand we see here is mainly in tumor macrophages, and these guys are mainly in macrophages, right? So what we're seeing here is macrophages and monocytes are dominating the expression in the APO ligand, right? Where the tumor epithelium is near zero. This is directly contradicts our prediction of the APO to trim, right? This is the same problem we had in the last one. We saw a signal, it made sense, cell chat told us it was right, and then we went a little bit further to investigate it, and it was wrong. And that's just the way it is. It's not always right. It's complicated, right? And what we're basically seeing here is instead of we're seeing the signal, but it's more of an autocrine signal coming back. And if you visualize it here, this APO network, it's really a monocyte to macrophage autosign, different things. So we learned about the pathway. We found things that made sense in the biology of our tumor. It just wasn't exactly what we expected. And that's how cell chat is. And we see here they're mainly to each other. But that's the problem with cell chat. It's hard, you know. And we looked at some of the pathways. We kind of saw that they didn't quite match for a couple of those either. And you can kind of look into this a little bit further, just to kind of see that a couple legs of the triangle, we did like a triangulation. I guess I can kind of tell you the result on that, where like basically 2 of the 3 legs failed. We had expression in myeloid and TREM2, particularly the macrophages. And then we had depleted inflammatory programs. Those kind of made sense, but the lipid metabolism leg ended up being absent from those results we tried to compare over. So we see here is that macrophages in this TMA are really a functionally suppressive inflammatory program consistent with its TREM2, you know, polarization, this M2 polarization, right? And this APO TREM2 autocrine loop is actually the real driver. Because the metabolic programming signature expected was not detectable the way we thought. So, and we have a small sample size. We're hand-waving a lot of this stuff with the sample size. You may be able to see a lot of this come out if you weren't doing something different. All right, so our final practice problem, and I'm going to let you do a lot of this because this is a pretty long problem here, but we're taking that odd module score and just giving you ideas of different things you can do with it. Some things work, some things don't. That's the point. You want to be super creative. You know, you can do canonical marker scoring for cluster annotation. You can do continuous cell state scoring that we talked about. You can apply the pathway projection. But you can also do things like take in transitional published signature scoring to connect your data to clinical outcomes. You can do rigorous score availability of your own gene sets to find out how many genes you actually should have in. I mean, you can do all sorts of things. The whole thing is you can make any kind of custom signature. Connect it to survival, connect it to the patient, connect it to whatever. Be creative, make your own signature and score it. And there's no holding back what you do. I mean, the sky's the limit. So we did a couple things to show you how to do things. We did a cluster annotation, which worked pretty well with our T cell, but it often is not the greatest thing to do with a wide, full clustering of all your cells. It works sometimes it doesn't. Sometimes it's better to have just the genes. Sometimes they cross over too much and bleed through and make complications. But for example of what you can do, you can just make your own gene signature. For each cell type, you have your gene signature. Then you score them. kind of like we did last workshop with the T cells at the end. And then we look and we just see which one's the highest in each cluster, and we say that's the column. And that's kind of what we did here. Long story short, this is what we got, and it was pretty good. You know, we got our CD8, CD4. We're actually backwards here. Actually, let's see. So CD4, yeah, they weren't backwards. These are right. Actually, I forgot the way they're flipped in our other thing. NK cell, nailed it. B cell, mast cells, tumor. Macrophages, monocytes are a little bit off. It should be more macrophage here, but it didn't do bad. And we could have gone through and iterated through signatures and done a better job, but mainly I just want to show you this is the way you could do it. 2, cell state scoring. We've done this already, right? But what you can do, go in papers and find their scores, and you can apply their signatures, and to be able to see how those affect your own thing, your own work, right? Look at your naive memory, effector activated. You can really look at all these things, all sorts of different kind of different states and trajectories you can look at. But I'll give you a chance to go through those here. They're really cool, and it really is just showing you different ideas and creative ways of using these. Pathway activity, well, we did that one like that with the FGCA. We just kind of looked at these hallmarks, and we saw these kind of like before, projected them back, kind of looked at them, and then we kind of compare the hallmarks by scores, by cells, you know what I mean? Kind of look more into that. We took a translational signature that's published, right? To be able to connect our data to clinical outcomes. And basically what we found there is this clinically validated gene signature that predicted immunotherapy availability. And it kind of had this signature, and then we were able to use the signature, go in and look, and kind of see and look into how well and predict the response of this patient based on the scoring of the signature. Another way you could use it. One thing you can do is, do I have enough genes? It's always like, do I put too few genes, too many genes? How many genes should I choose? Well, you can go through permutation and gene set design and kind of look through each one of your gene sets and find out very robustly, very statistically, how many you should have in there. And finally, you can, I guess it's the last one I have, and you can kind of have better, stronger belief they're going to work, right? So that's kind of the end of that. The idea is you can just take anything you want and make a cool add module score out of it, and they're very creative. And I think all of these problems, if you'll kind of look through them more in depth and really read through them, I think it'll add a lot of value to the lesson and what you can get out of it. And then here, like I always do, some common pitfalls, additional resources, foundation papers, and that's pretty much it. Workshop 3 complete. I think we have a pretty good understanding now of foundationally to go from our raw counts to our cell type annotations, and then take those to further move down to our downstream effectors and analysis and then clinical outcomes. I hope you've enjoyed our path through through single cell RNA. This is a really cool field. It's still very, very important and growing. And on our next couple workshops, we're going to apply this to actual spatial transcriptomics. We're going to use a Visium data set and move over to there and be able to work you through some very, very cool spatial transcriptomic workflows. And I look forward to seeing you then. Thank you again for being here.